Okay, great. Everybody ready, Jim?
Yeah, we're good.
Okay. Well, welcome everyone. Again, I'm Dennis Sikowski, Science Director here at Hudson River Foundation. Thanks for coming. This is the last seminar of the spring, our spring series, and we'll be starting again back in the fall. And I will ask that if any of you, both here in our office or any of you that are on the webinar, have any suggestions for talks that you may be interested in next year, please drop us a note. We would love to get your suggestions. Today, we're pleased to have Jay Field with us. Jay is a scientist with the National Oceanic and Atmospheric Administration. Jay flew out from Seattle to be with us, which we're very appreciative of. He's been in the Office of Response and Restoration, I like to say a very short time, only since 1986. And he's an expert and has a lot of experience in dealing with contaminants, and particularly the fate and effects of contaminants like PCBs. And it's his expertise and NOAA's interest in natural resource damage claims has brought Jay into direct contact with a number of large Superfund sites, such as Passaic and Berries Creek, Duwamish River, Kalamazoo, and I think probably, if not his, and Newark Bay, and if not his favorite, probably our favorite here, the Upper Hudson GE site. So as I think most of you do know, that this is the last year of scheduled, I'm not sure I'll say this right, the last year of scheduled dredging in the Upper Hudson. And it's, I think, very appropriate that we would like to reflect on what's going on and what may be happening in the future, and an assessment of how well that project has gone and will go into the future. So we've asked Jay to come in and talk specifically about some of his work. So the title of Jay's talk is Revisiting Model Projections of Lower Hudson River Fish PCBs Using Model, Emulation, and Recent Data. And just 1 second before, I just want to remind folks that are on the webinar that hopefully you can hear us and see us here, and you'll be able to hear Jay. And at the end of the talk, you'll be able to write in questions for Jay that we'll be able to take after the talk. So Jay, thank you.
Thank you, Dennis, and thank you all for coming and for listening online. My wife looked at this title and said only Hudson River aficionados are going to come to that talk. So I appreciate your coming to this. I'd like to, I thought it was kind of a catchy title myself. Acknowledge my co-authors, John Kern with Kern Statistical Services and Lisa Rossman with NOAA's Office of Response and Restoration in Seattle. I mean, it's in New York. I'm in Seattle. She's in New York. So what I want to talk about today are some of the model projections that were made that played an important role in EPA's record of decision in 2002 for the Hudson River PCB Superfund site. Subsequent to the ROD, a lot of data was collected for remedial design that suggested that the rate of recovery of the surface sediment was overestimated by the models. And so we are using an approach of model emulation to update these original mechanistic models with the new data that was collected on the remedial design. So models are extremely important for these large Superfund sites. It's really the only way to predict what the impact of a decision is going to be in the future. And these kind of models are used throughout the country and on large Superfund sites. And because of the extensive data that we have, it gives us a rare opportunity to actually revisit the model predictions and see how well they did. So we want to look at two questions. What's the impact of this new information on the mechanistic model projections for recovery of fish in the lower Hudson River? We're focusing on the lower Hudson River because the fish were expected to recover much more quickly than the upper Hudson River. And also, what's the implication of our findings for use of these kind of models in comparing remedial alternatives? In terms of what I'm going to cover today, I'm going to give a brief background on the Superfund site in the Hudson River, the selected remedy. I'm going to review the findings from the post-rod sampling, give you a quick sketch of the mechanistic modeling that EPA used in the decision-making process for the upper Hudson and the lower Hudson, and describe how we emulate those models and apply the emulated models to look at the impact of the findings on the model projections for lower Hudson River fish. The Hudson River PCB Superfund site extends about 200 miles from the village of Hudson Falls down to the Battery in New York City. The upper Hudson River is separated from the lower Hudson River by the federal dam at Troy, just north of Albany. The upper Hudson River is about 40 plus miles. The lower Hudson River is 150 miles of tidal estuary. The EPA remedy addressed the part of the upper Hudson River between the former Fort Edward Dam and the Three River sections down to the federal dam. The two GE plant sites up at Hudson Falls and Port Edward are being addressed under a New York State remediation process. So the remedy that was selected for the upper Hudson River, all the remediation is taking place in the upper Hudson River, referred to as REM 310 Select. It included upstream source control, which I mentioned as part of a New York State process. And it included target cleanup levels that had two components: a mass per unit area, measure of essentially PCB inventory, and a surface sediment level that represented the concentrations in the top 12 inches of sediment, as defined by EPA. The target cleanup levels for river sections 2 and 3 were about 3 times higher than the target cleanup levels for river section 1. And throughout this presentation and throughout most of this project, PCBs are looked at as trichloro PCBs, tri-plus PCBs, the trichlorobiphenyl and higher chlorinated congeners, primarily to be consistent with historical analytical data, but also because fish predominantly have tri-plus as total PCBs. They don't accumulate the mono and dichlorobiphenyls very much. And I should mention that dredging was expected to start in 2003 and extend to 2000 and be completely done by 2010. It actually started in 2009 and is expected to be completed at the end of this year. So during the sampling for remedial design, the sampling protocol was a systematic sampling design, basically an unbiased sampling design. It extended for a number of years. We're looking at the data between 2002 and 2005 because that's when most of the systematic sampling was collected, was done. In River Section 1, essentially the whole section was sampled. In River Section 2 and River Section 3, the cohesive sediment or the fine-grained sediments were targeted almost exclusively. There were over 8,000 cores that were collected during this time period, with PCBs measured in the top 2 inches of each core. And so we're using this data to look at mean concentrations for the individual river sections. We're assuming it represents the year 2003, and the 5 centimeter samples are comparable to the 4 centimeter samples used in the modeling. Used in the modeling. So this is a slide showing a comparison of the mechanistic model predictions for mechanistic model predictions with the post-raw data. The bar chart here shows the mechanistic model predictions for the mean and upper bound, and whatever that color is, tan olive, on the left for each river section. The sections are on the x-axis, and the concentration tri-plus PCBs on the y-axis. And the blue bars represent the mean and upper 95% confidence interval from the remedial design sampling. And you can see that the concentrations in the surface sediment exceeded the mean concentrations by a factor of 2 to 3, and the upper bound of model predictions by throughout the upper river. This second panel shows the estimated post-dredge concentrations. Once again, the mechanistic model predictions are in the bars on the left, and you can see that the model predicted that the concentrations throughout the upper Hudson River would be 1 ppm or less tri-plus, and the data from the post-rod sampling, remedial design sampling, was 3 to 5 times higher than the model predictions. And so in the rest of this presentation, we're going to use the post-rod sampling for sediment, pre-dredge and post-dredge, as the updated information to use in our emulated models. We also wanted to look at the rate of recovery of the sediment. So we have the remedial design data that we're assuming represents 2003, and GE in 1991 conducted a transect survey throughout the upper Hudson, and we're using the concentrations for cohesive sediment to compare with the remedial design data, and fitting a simple exponential decay model function to the values, and we get a mean decay rate of 1.3% with the confidence limit from 0.1 to 2.6%. We're rounding the upper bound and using 3% as the way to compare it to the mechanistic models that essentially had a rate of recovery equivalent to 8%. So you can use that in the models. So in summary of the post-rod findings, the sediment concentrations in the surface were much higher than predicted by the mechanistic models. The rate of recovery was slower than predicted by the models. And I didn't show you this, but this is from EPA's 2009 evaluation report, that the load from the upper Hudson to the lower Hudson was greater than predicted by the models, and showed little evidence of decline. So this is another reason we're interested in looking at the lower river fish, because obviously that's going to have an impact on them. So, as I mentioned, we're going to be, these findings have a lot of importance for the lower river, and we didn't have the option of rerunning the original mechanistic models, at least in part because of the cost and effort that would be involved. So we turn to model emulation, which gives us an alternative approach to efficiently condense these complicated models into a simple and easy to use model, while at the same time still maintaining the underlying relationships that are within the mechanistic model. It gives us an opportunity to consider alternative scenarios, and this kind of approach has been used effectively by large numerical ocean models and climate change models. So this is a cartoon of the mechanistic model. Basically, it all starts with the surface sediment, and the surface sediment through a number of model subcomponents predict the surface sediment in each river model subsection in the upper Hudson. And from that, PCBs and water for each subsection are projected. It includes an upstream input from the source from the plant sites, and that was factored into the model. And the output from the upper Hudson models was then used to input into lower Hudson models to predict PCBs and fish for 4 species at 4 locations. So what I want to point out on this map is that the load for calculated from river section 3B, Waterford, is what is was used by the mechanistic models to input into the lower Hudson River models. So, and the output from the lower Hudson models, from the upper Hudson models, was then factored in to put into the Farley lower estuary model to estimate water and sediment in the lower Hudson, and then the output from that was used in EPA's fish ran food grab model for white perch, largemouth bass, brown bullhead, and yellow perch at the 4 locations that you see here from just below the dam down to River Mile 50. So we're going to be looking at 3 of the scenarios, that mechanistic model scenarios that were part of the record decision, or evaluated in the record decision. M&A monitored natural attenuation. It assumed an upstream boundary condition input into the Thompson Island pool of 2 nanograms per liter by 2005. That appears to be a pretty good assumption. The selected remedy that I've described earlier, and they also, the other dredging remedy that they considered was a full section removal in river section 1 and 2, referred to as the 003 remedy. And it was and as I mentioned, dredging was expected to start in 2003 and be done by 2010. So these throats show output from the mechanistic models for the 3 remedial alternatives. The panel on the left is the cohesive sediment from river section 2. The panel in the middle is the water concentrations for Waterford, river section 3B. And the panel on the right is the concentrations in fish, white perch, from the Albany Troy location. And what you can see is that the trajectories look similar in all 3 matrices. And particularly, I want to point out that and the black line is the M&A, the blue line in the middle is the selected remedy, and the red line at the bottom is the 003 full section removal. And what I want to point out is the very small difference between the predictions for the selected remedy compared to the full section removal. And that was cited in the record of decision as being important for not moving forward with the 003 alternative because of that small difference. the 003 alternative would have essentially doubled the size of the remedy, and it was rejected, at least in part, because it didn't provide much improvement according to the models. Also extremely expensive. So the model emulation approach that we're using, we're developing statistical models to reproduce the mechanistic model projections for sediment and water in the upper Hudson River and for lower Hudson River fish. And we're doing this for the moderate natural attenuation and the selected remedy. And then once we've developed these models, we can apply the new information from the post-rod sampling to assess the impact on the predictions the models made about the concentrations in lower river fish. So the model emulation for sediment, we're essentially, the initial emulation uses directly the model output from the mechanistic models. When we get into the updated information, we're looking at the sediment in a couple of ways. One is using the proportional change on an annual basis in the model projections, and also we're using an exponential model to describe the data. For the water, we're estimating the water concentrations from the sediment using a nonlinear regression for each subsection, and then we're taking the water concentration from Waterford using strictly a linear regression approach to predict the PCB concentrations as modeled by EPA. So this just shows you that the complex mechanistic model is being basically described by a nonlinear regression for predicting water as a function of sediment and a couple of other variables. And then the output from the water is being plugged into a linear regression for fish concentrations. So this is what the water emulation looks like. What you see in this plot is the mechanistic model on the predictions on the x-axis for M&A and the selected alternative selected remedy, and the emulated concentrations on the y-axis. The dotted line represents the one-to-one line, and each of the different symbols and colors represents a different river subsection. Particularly, if you can see them, the black triangles, which represent the concentrations in river section 3B, Waterford, are almost right on the one-to-one line, and that's important because those are the values that we're using to plug into the model to predict fish. So this shows the emulation for fish. Essentially, we're using the mechanistic model output for water concentrations at Waterford against the fish concentrations predicted by the model. And this is for the 4 species that were modeled from the Albany-Troy location, River Mile 152. And you see that the linear regressions do a very good job of describing the relationship in the model output. The other locations look pretty much the same as this. So just as a frame of reference, throughout the rest of this presentation, we're going to use white perch at Albany-Troy as kind of the model that we're going to evaluate. And here we're comparing the white perch concentrations as predicted by the model, the mechanistic model, with the concentrations in the other 3 species. The dashed line is the one-to-one line. And as you can see, largemouth bass were predicted to have higher concentrations than the white perch, and the brown bullhead and yellow perch were predicted to have lower concentrations. Similarly, now we're comparing the other 3 locations to the white perch concentrations at Albany, Troy, and as you would expect, the concentrations were predicted to go down as you go downriver from the dam. And the relationships are very consistent and this is all the mechanistic model output here. We're not doing anything to it. So we also wanted to look at the rate of decay. So this is the mechanistic model output for fish. We could also do the same thing for sediment. This shows that the selected remedy in the blue and the M&A on the top in the red squares. The lines represent an 8% exponential decay model, and you can see that the exponential decay model captures the model output pretty well. So that gives us the ability to apply an exponential decay function to describe some of the model output. So we're going to look at several emulated model scenarios. We're going to, and the abbreviations in bold represent symbols you're going to, or abbreviations you're going to see in some of the later figures. For the original model projections for M&A and the selective remedy, we're going to be looking at the same M&A and the selective remedy with updated sediment concentrations based on the post-rod sampling. And we're also considering an alternative scenario that wasn't considered in the record of decision. We're applying, essentially applying the same river section 1 criteria for mass per unit area and the surface concentration to river sections 2 and 3. And we're going to be using that with the updated sediment concentrations. That's not nearly as extensive as the full section removal 003 that EPA considered, but it's certainly more than the selected remedy. And for all of these, we're going to be looking at an exponential decrease of 8%, which is what the original model was, result was, and the updated 3% that I described earlier. So this slide shows a model year starting at 2010 on the x-axis and the concentrations in white perch from Albany, Troy. The hollow symbols, the circles and squares at the bottom, represent the original mechanistic model predictions for white perch, and the solid red square and blue circle at the top represent M&A, and the selected remedy using just the updated data, same, no additional exponential decay applied, so we're using the same decay that the model predicted. So what you see is not surprising. The concentrations in the white perch are projected to be higher and extend higher and longer than the model predictions, and they also have a wider difference in concentration. So, one of the ways that was used to evaluate the fish concentrations was to compare them to human health risk criteria that EPA established in the record of decision. And this is the human health remedial action objective that was used. And they used 3 values. The goal of 0.05 milligrams per kilogram PCBs represent a half a pound meal a week. 0.2 milligrams per kilogram PCBs, which represents a half pound meal per month, and 0.4 ppm PCBs, which represents a half pound meal every 2 months. These are in total PCBs, but as I mentioned before, and we're comparing it to tri-plus PCBs predicted by the model, but as mentioned before, almost all the PCBs in fish are as tri-plus. So EPA in the record of decision said that the remediation goal was likely to be reached in the majority of the lower river fish after the completion of the remedy, or at some point after the completion of the remedy. So this is a complicated slide. This shows the hollow circles represent the original model predictions. This time we're using an exponential decay model to describe the results. We're showing those in the hollow circles at the bottom. And the hollow circle is the selected remedy. The solid blue circle is the updated information. So what you see at the bottom, you saw previously as in a slightly different form. So that's at 8% decay, the original model predictions for the data, for the sediment, and the three solid symbols represent a 3% decay with using the updated data. And once again, we're looking at white perch from Albany, Troy. So you see a number of things. You see a revised estimate of the amount of natural attenuation, which is much higher than any of the other alternatives. The selected remedy is in blue is considerably higher than the model projections. And our alternative scenario using essentially the 333 option is considerably lower than the selected remedy, but still quite a bit above what the model had projected would be reached after the completion of the remedy. The two lines on the bottom, the dashed line at the bottom is the 0.05 level, and the dashed brown line is the 0.2 threshold. I didn't show the 0.4 just to keep it from getting too busy, but you can see that the initial projections were heading into those areas very quickly, whereas the selected remedy is not expected to get there for quite a while, according to the, if we're using this updated information and an updated exponential decay rate. And the other thing that I think is really important to note on this slide is that there's a large difference, a much larger difference, apparent difference between the model outputs when you use the 3% decay than when you use the 8% decay, which is the difference is perceived to be much greater and is much greater in terms of concentration. So this is another way of looking at the output, and here we're just looking at the number of years, which is the y-axis, to reaching the thresholds of 0.4 on the left and 0.2 on the right. We're comparing REM1, the original remedy, with the original data from sediment data predicted by the EPA mechanistic models. The selected remedy, REM2, applying the updated sediment data, and REM3, the alternative option that we included for comparison using the same target cleanup levels for river section 2 and 3 as in 1. And both the blue bar represents a 3% decay, and the tan bar represents an 8% decay, so it gives you a sense of how different those outcomes would be depending on how fast the model is predicting the decay of the surface sediment. You can see for the selected remedy for 0.4, they expected the concentrations in white perch at Albany Troy to be below the 0.4 threshold by the time the remedy was completed. And also the panel at the top has the actual numbers estimated, number of years estimated. We stopped the emulation at 25 years, but extrapolated out. So you can see that the selected remedy at 3% was over 4 times as predicted to take 4 times as long as the same remedy at 8%. And the alternative that we considered is obviously a lot lower in projections, and but still doesn't reach the level of the thresholds throughout the period, the 25 or the even closer, unless you're looking at 0.4. So, and you also need to keep in mind that the other species at other locations will have slightly different values. As you go farther downstream, the thresholds will be more attainable. So this is almost the worst case. Largemouth bass would be the worst case, but this is the example we're showing. Oops. So in summary, for the model emulation, we were successful in reproducing the mechanistic model output for sediment, water, and fish PCBs under M&A and the selected remedy. This made it possible for us to update the sediment concentrations and also update the estimated rate of sediment recovery to revisit the projections that were made by the mechanistic models for lower river fish concentrations. It also gave us an opportunity to consider different scenarios. I showed you one. Other advantages that I didn't have a chance to talk about much, but these models are really useful for doing the kind of statistical uncertainty evaluations that the large complex models really can't. And so you can look at different outcomes and get a sense of where the most uncertainty is, and it's also be a useful tool for more accurate calibration and validation of the models. For the Hudson River fish, the recovery of the sediment, the sediment surface recovered much slower than predicted. Same with the fish. The concentrations in fish were slower to go down than the original model projections. Applying the alternative, more aggressive remedy than the selected remedy would reduce the time to reach the thresholds that were established, but still longer than originally predicted for the selected remedy. So these findings also have some implications for the use of these kind of complex models in decision making. As you saw in several of the slides, the overestimation of the rate of natural recovery tends to minimize the difference among different remedial alternatives. If you don't have an accurate estimation of the rate of natural recovery, you have no good way to really evaluate the comparisons of remedial alternatives. So in order to get a good rate of recovery or to ground truth your model, you need to have solid baseline sediment data collected systematically so you can make temporal comparisons of sediment concentrations. Otherwise, your comparisons of remedial alternatives can be highly misleading. And as I mentioned, the model emulation approach can be very useful in reducing and understanding the uncertainty that you're dealing with. So in conclusion, the original models were overly optimistic about the rate of recovery of surface sediment under M&A and the selected remedy. Attainment of EPA's remedial action objectives for fish in the lower Hudson is likely to take much longer than predicted. And the additional removal of PCB contaminated sediment from the upper Hudson is probably needed to achieve the reductions in lower river fish concentrations that were anticipated in the record of decision. Thank you.
Questions?
So, what went wrong? Were the concentrations in the sediment just higher than expected, or was just the rate of natural attenuation much lower than expected?
Could you just repeat the question for people on the left?
So the question is, what went wrong with the models that they predicted the recovery to be much faster? If that's fair. Well, that's an excellent question. There's a lot of things that go into that. The models, the output of the models is the rate of recovery, and that's not a parameter that goes into the models. So, there are a lot of things that could affect that. Certainly, the data that they were starting with could affect it, but also the inner workings of the model could also be biased in a way to get a faster recovery. One of the things that we think about is all these models, both EPA and GE use similar models in terms of the basic structure. One of the common assumptions was that your mixing depth was 10 centimeters. GE actually did a sensitivity analysis of that parameter, and what they found, they used 5 centimeters, 10 centimeters, and 15 centimeters. And they reported that the 15 centimeter actually described the data a little better. And because it was, because there's a nonlinear relationship, you're much more likely to have a problem if you underpredict the mixing depth. So, but they stuck with 10, I guess, because they didn't see as much of a difference as to make them change. But that's kind of one of the things that I would look at. But it's really difficult to tell. That's probably something for the modelers to try to figure out.
I know that we had a lot of success of the dredging on the fish tissue concentration, and rightly so, but there are other goals in the ROD. One of them is reducing the inventory of PCBs that'll become bioavailable and end up in New York Harbor as well. How is that being measured as a success? How are they measuring that, whether it's going to happen or not?
Well, that's, Ed, you could probably answer that better than I could. So the question, as I understand it, is how are they measuring the reduction in inventory, presumably in the upper Hudson?
Especially if the models of the coming over the Troy Dam and the fact they did, uncover deeper portions of PCBs than expected. How is that being measured for success as achieving the goal?
I think, they've identified the areas they're removing. I don't know what the post-remediation sampling plan, or if there is one yet, but that would be the best way to determine what's left behind. But in order to do that, you've got to sample not only the area that you removed, but the surrounding area. And as far as I know, there are no plans to do that, but there may be.
We have a question up there, Jay, from Manager Green.
So we didn't really have a good way of evaluating the 003 remedy, but obviously would remove a lot more inventory than any of than the selected remedy or the alternative remedy scenario that we looked at. It likely would have a greater increase of greater effect on the decline of fish concentrations in the lower river as well as the upper river, but we have no way of really quantifying that.
Is that just a methodological challenge to emulating another remedy?
Well, we didn't really know what how to project the 003 remedy, so because it was a full section removal, it just seemed a little artificial to try to do that, so we went with something that was easier to kind of conceptualize.
Yes, so the model considered the pathways where the fish consumed the PCBs and it went through his body and he discharged some of it. This may have been later uptake by other fish in the food chain. Was that in any way considered the PCB pathways?
Well, the mechanistic models developed by EPA were very comprehensive in terms of evaluating the food web. So they have a lot of complexity. I don't know if they considered that particular element, but they certainly are considering the food web. And all we're doing is just basically modeling the output of the mechanistic model. So whatever, we take the starting point and the end point, and that's what we're looking at. So I don't know if that answers your question, but.
I may have missed this or I just may not understand it very well, but could you explain again why using 3% rate of decay versus the 8%?
Well, the question was why we use 3% rate of decay rather than 8%. Well, the 3% was the upper bound of our empirical estimate, just based on the data we had. We don't have a lot of, to make an estimate of the rate of recovery, you really need good data, systematic sampling, so that you can compare what was found. I mean, the data set from the remedial design is a really solid data set, but using the data from the GE Transect survey in '91 provides us I think the only systematic sampling that was done early enough to use. I don't know how solid those comparisons are, but so that was our estimate. We actually estimated that the rate of decay would be lower. The 8% was what the mechanistic model output resulted in, and based on the finding that the sediment concentrations were a lot higher than anybody predicted, it kind of points the finger at the rate of decay was way too fast.
I have a question about systematic sampling. What do you exactly mean by that? Just throw a grid equally spaced everywhere and then 5 years later go to those same locations, as opposed to, we want to focus on certain areas that we find higher concentrations or maybe subject to erosion?
Well, that's a question for a statistician, but the best way that I could describe it would be to you have to stratify by the type of area, and then you do identify areas. So you would be comparing a lot of samples from an area as opposed to individual samples. So you'd be trying to define the area statistically, compare it to another, the same area, sampled again. Does that help? My co-author could describe that in a lot more detail.
So in your, I think you called it scenario 3, that you guys ran. Did you also make an estimate of what kind of volume of removal that would be, like how much volume that would be, and how long that would take if that were?
We didn't estimate volume. We don't really have the information to estimate volume. We did make a crude estimate of area, and the area from that would be about 200 acres compared to the almost 500 acres in the current plan. And just for comparison, the 003 was projected to basically double that 470, so it's almost 1,000 acres.
My question kind of related to that. Is it possible that there was more PCBs in the site than was initially discovered? And when they started to remove, there were reservoirs that now appeared that no one had identified before that seeped in, so that when you get the levels as high as they are, could that be a consideration? In other words, it was under count, and now it's showing up in the street.
Well, I guess if you're asking, was the surface or the upper part of the sediment not well sampled before, and now when we're doing this extensive sampling, we're finding these concentrations, or are you saying that it's coming in from?
Well, just experience on and upland stuff that, chromium from the water plant, for example. That stuff just keeps coming out of sites that you had no idea was there once you remove some of the initial soil. But that's upland out in stream. I'm just saying, could that same phenomenon be evident here, that they just dumped so much stuff that you could get the top of it to look at initially 10 years ago? Well, now as you took that top of that stuff, just kind of leaked back into the testing site. I don't know.
Well, they did extensive sampling back in the late 70s. They did additional sampling in New York State in, I think, 1983, 84 timeframe, and then there was the sampling during the reassessment. And I think, they covered a lot of they covered Thompson Island Pool very well. River Section 2, probably less well, and River Section 3, a lot less well. So there are probably areas that weren't sampled well. One of the things that you can look at is you can look at the original hotspot description in the EPA reassessment, and when you look at those defined hotspots compared to where they're actually doing their dredging and the dredge prisms that have been identified, some of them are very different. So it's like either concentrations, sediment moved, which is possible, or there was just poorly mapped to begin with because there was insufficient data.
Got another question, and maybe you could read it too.
Okay, the question from Jonathan Clough was, I would assume that the original mechanistic model included data about the ratio of sediment depth versus surface sediment concentration. If that ratio changes due to remediation, for example, the surface sediment has higher concentrations than deeper sediment now, couldn't this have the potential to bias the derived metadata model or the emulated model? Not sure I understand the question, but the ratio, once the removal is complete, the post-removal sampling documents that there's essentially no inventory left. So I'm not sure how that, does anybody understand this question?
Is he asking how you're taking the sampling? Does he understand how far down the cores are going? 6 inches.
The original mechanistic model had a three-dimensional picture to some extent, because as you point out, a lot of the cores are looking all the way down. They're trying to find the bottom of the contamination.
But in the emulation, you're just making an assumption about what the surface would be in the areas that have been remediated.
Correct.
You're using the existing data outside
Right.
of those.
Right. So we don't
it
change
shouldn't
the.
really change.
Yeah. So, and the assumption we make after the remediation was, oh, good. Okay. I guess, I think the answer is, so he's asking if, according to John Kern, he's asking if the emulator is equally applicable pre and post remedy. I can't see why it wouldn't be, and I'm not quite sure how what he's got there would change that. But maybe I'm missing something.
So if you use the center of your decay range rather than the most optimistic number, and it was probably around 1.5 instead of 3%,
Yeah,
how much further out would that push those curves?
it would, that's essentially cutting the decay rate in half from what we showed. It wouldn't be as great a difference as the difference between 3 and 8, but it would be a big difference.
I mean, it seems like a more likely.
And one thing I should point out is that as you get down to these low concentrations, the estimates of how long it's going to take, the uncertainty gets larger as you get very low in the concentration range. So, is that?
Yeah, it is. So from a sensitivity analysis perspective, was density, decay rate versus surface concentration, which one had sort of more leverage over your model output?
Well, probably they both do. And I showed you the one with just looking at the sediment, and you saw the difference from that using the 8% decay, but the decay rate has a huge influence on the rate of recovery, because that essentially defines what the concentration is outside the dredge prism, and how fast that is going to change. Okay.
Question for you, Jay. The bioaccumulation model basically is a ratio to the sediments that the animals are exposed to. Since the data show that the original premise of the bioaccumulation model is based on surface sediment concentration that was incorrect, the ratio of sediment to tissue changes. How did you account for that?
Well, we're not looking in the lower river where the model takes into account water and sediment, and probably the water plays a very important role because of the amount that's coming over the dam. So we're not, the food web model that was used, both the Farley model and the fish ran food web model, are looking at the concentrations in both media and using that as a way to model the concentrations in fish. So it isn't just a strict BSAF type approach, sediment to fish approach. It might be, you might look at it that way in the upper Hudson, but we were focusing on the lower Hudson.
If I remember correctly, I think the EPA is currently taking water measurements of PCB levels in the Hudson where they're dredging. And so I'm just curious if they're going to use that more current data and perhaps some other data collected by other researchers on fish tissue concentrations or sediments to either verify your models that you have since they were done so early, or to, reevaluate and update your models.
Well, keep in mind that the question was have we thought about using water monitoring data to update the models? And keep in mind that, we're emulating the mechanistic model output. We're not starting from the beginning. So, that could be factored in, but that would affect, since we're predicting the water from the sediment, we'd have to be able to, basically calibrate to that or change our model. So we don't really have any easy way to do that. We can just look to see how they compare to what the models predicted or what the emulated models predicted.
So then I guess just to follow up on that, have you done the comparison and how well do things align to which are the scenarios?
So, so the question is, have we compared our emulated model results to real data? The way we would try to do that would be to look at the fish concentrations in the lower river, particularly prior to the initiation of dredging in 2009. And there's a complication that we recently became aware of that makes that difficult to do. Under the leadership of Ron Sloan for New York State, the fish monitoring program developed very standard protocols for how to collect and process the samples they collected. And in particular, they had a standard fillet approach that attempted to minimize the number of cuts that were made, both to make it easier for the processor, but also to have consistency in the data. When GE started the monitoring, essentially took over the monitoring program, the baseline monitoring program in 2004, and then the remedial action monitoring program. At some point, and we don't know when, maybe EPA does, but GE changed the processing to no longer include the rib cage in the fish fillets, and EPA had GE do a study last year or the year before, looking at largemouth and smallmouth bass and taking fillets with the rib cage and without the rib cage. And the preliminary analysis from those comparisons suggests that the fillet concentrations using the GE's revised fillet method underestimate the wet weight PCBs by about 30%. So we're not sure. We're not sure, one, if this applies to all fish species or whether it applies to when it actually started. So it's hard to make those comparisons. We do know that the white perch are elevated, even with the current concentrations are elevated relative to the model predictions, but we don't know how much.
Jay, you mentioned with the emulation model that you can go back and take a look at the uncertainty, I guess, in certain outputs. Would that include fish tissue, water concentration, all the various types of things?
Yeah, it could.
Because getting at it, I mean, you made the point that the assumption that there was a continual decline in concentration and therefore load downriver was something that was not really occurring prior, I guess, to the rod. I mean, we picked that up in our carpet program as well. And sort of adding on to what these folks were asking before, I mean, can you look at that, the water concentration in any detail to see whether, it just seems to me that the model's getting that wrong, getting water concentration wrong, and is anyone looking at that, or can you look at it through the emulation to look at what's causing that uncertainty?
Well, we'd have to have the data from the same place, measured in the same way, which is is a problem with water data. Ed probably knows this better than I do about where the water collections are coming from, but it would be a challenge to incorporate, the water data, but it probably could be done, but it's not.
But I guess it's just maybe Ed can comment, it just seems to me that if the emulation is essentially mimicking the mechanistic model, and if the water concentrations aren't right, I mean, wouldn't it even be a worse error than you're even showing, with having a lot more PCBs coming out of the sediment and moving downriver, and increasing exposure to fish?
It's possible. Off the top of my head, we don't have any easy way to look at that question. I mean, there are a lot of questions. What we're doing is a pretty simple-minded thing. We're basically mimicking the mechanistic model output. You can bring in a lot of factors and make it a lot more complicated, but we basically wanted to look at what the model predictions were and how we thought they would change if they had more current information. But there's lots of ways to add, bring in that more current information. Obviously, the more information you have on the different components, the stronger any model is going to be.
With another comment.
Oh yeah, okay, so what John Claude was interested in was whether there's a post-dredging transient condition in the surface sediments that would cause a disconnect between the mechanistic model and the meta model. Well, I guess if that was considered in the mechanistic model, then we would have essentially have captured that in our emulation of the mechanistic model. We didn't directly consider it. We did see something when we were setting up the emulation that made us look at the data immediately post dredging a little differently than the rest of the data. So we actually it looked like in the mechanistic model there was some element of resuspension and resettling in the mechanistic model, and we tried to capture that in the emulation, and I think we did a pretty good job of doing that in terms of the mechanistic model predictions.
Okay. Just want to understand what I think I understand from this. So, because of the surface sediments in the upper river were underestimated, in terms of the impacts on the lower river, the loads in the lower river are underestimated as a result. And so, because those loads are underestimated, there's a longer-term impact in the lower river. And I would throw this back as a question to you that the original model wasn't just calibrated to the sediment. It was calibrated to sediment, surface water, and fish. It was flooded all together, and I'm an empiricist. So as far as I'm concerned, we use control dials, so its model is the trajectory. Okay? And now we have a different set of initial conditions, if you would, that get set the dials differently for, and that's your point. Okay? I don't disagree with that. But that also means that trying to get the water, if you now use your readjusted for sediment model, your water column loads are now, you probably, if you were to do a high gas, so to speak, go back and look at what your model would do in terms of water column loads historically, I'm guessing you're going to overestimate it because the model had some sediment to water exchange rate that generated the water column condition based on what it thought surface sediments were. Now you've got a different surface sediment condition, so you've got a different exchange rate. To use that same exchange rate with a higher concentration, you're going to get way too much load. So I think that's an important thing for you to consider. I know you've done that.
No, we haven't done it, and we're as I mentioned before, what we're trying to do is basically a simple-minded approach to evaluate the model predictions. What you're talking about and what some of the other questions are driving at are basically redoing the original model and making it better. And one of the advantages of the this simple model emulation approach is that it lets you ask those kinds of questions, and but it doesn't, it doesn't replace the more complicated model in terms of how it looks at the processes that are going on. So, we can't, we can do some simple stuff, but we're not going to turn it into a $5 million model.
After the peer review panel, to his point, the scientists on that independent panel recommended that the model be looked at, and I know that General Electric and EPA were working on it for about 6 months. Were you guys a part of that process? Did that help inform anything?
No, and as far as I know, GE is continuing to model, but I don't think EPA is involved anymore.
Well, no, they're not doing a new model anymore.
But the basic point is still, you can recalibrate the same model, put stainless steel knobs on it, and come up with a new predictions. But you're still working with the same data set. You have a little more information to ground truth it, but you still have you still have some potentially properties of the model, processes that are built into the model that may affect the outcome. So unless you have post-modeling data to evaluate how good their predictions are, as far as you could say, well, it's still the same model.
That's correct. Okay, well, again, thanks everybody here in the room. Thanks for the folks on the webinar. And stay tuned. We'll be posting a new schedule of seminars.
Are you going to close this presentation?
If we get Jay's permission, we will have it not only posted, but the video will be up there. So you can watch it over and over again.
I'm not going to watch the video. The slides.
Okay, thanks. Thank you.
That was great. Terrific.